slide in large format. Great, thanks.
Well, thanks so much for inviting me. I've really enjoyed all the talks so far, and I'm really looking forward to this week. My talk is in some sense about how far one can get without thinking too much about genetic data, I think. So I think I do what Hans Metz once referred to as meso-evolution, so not micro or macro, but somewhere between the two. And I've been really thinking a lot about, of course, we all do species interactions, but particularly about the remarkable success in predicting outcomes for host pathogen interactions. So for example, we've shown tremendous success in predicting the transition from annual to biennial dynamics as a result of changes in birth rate for measles. We've also, moving from the ecological to the evolutionary, developed really nuanced understandings of what happens to myxomatosis following introduction into places like Australia in terms of the evolution of virulence. So there are these sort of startling successes in understanding and projecting species dynamics around host pathogen interactions, possibly because this is a really tight interaction, so it might be one that's easy to unpick. But there's a kind of surprising aspect to this, which is the fact that that interaction also must happen across the immune system, which my friend Brian Grenfell once referred to as possibly the most complicated thing in the galaxy, with consciousness perhaps being alongside it. So it's kind of surprising thinking about why, how do we start thinking about the evolution of immune systems in this context? How do we fruitfully move forwards on these themes? And I was just going to run through two examples of how I've been thinking about tackling this from the context of within host dynamics, but also thinking about population level patterns to try and think about how this incredibly complicated integrative system works and shapes the patterns that we see. And I'm an ecologist by training. So of course, the first thing that if you're starting at the scale of within host dynamics, perhaps the first thing that springs to mind is a set of different ecological questions and a set of dynamical questions. And I think one of the reasons that the immune system is so hard to probe and understand is because it is an inherently dynamical system. We recently wrote a paper with Andrew Graham, who I think is also participating in this conference, thinking about what core ecological models might contribute, in particular to thinking about SARS-CoV-2. As we all, I think, unfortunately know too well, the serial interval of SARS-CoV-2 is about a week. The R0 is somewhere between 2 and 3. So that means that at a minimum, cases are doubling every week. You can apply exactly the same logic using a kind of SIR framework to how the virus is spreading within people's lungs. And estimates from Alan Perelson's group have estimated that the R0 is around 8, the serial interval is less than a day. And so you get this absolutely explosive growth inside people's lungs. And I think that's a useful way of thinking about it. There's also ways in which it looks like the peak titer doesn't vary much amongst males or females or people with more or less severe symptoms, despite the fact that we know that the female immune system reacts faster and seems to be more effective at slowing early growth. One way you might get around this is by thinking in terms of the immune system being some kind of predator that's responding to the density of the pathogen. And so you'd end up at the same level through this dynamical interaction. Of course, this is a grotesque simplification. We know that the immune system is not like a simple predator. It's much more like a huge collaborative swarm of items and elements and players. And people have done lots of lovely work suggesting that collective action might be really important in this setting. And I think one of the most kind of useful ways in which an ecological perspective immediately springs to mind when thinking about SARS-CoV-2 is the fact that we get such incredibly different health outcomes in apparently really similar individuals. And this echoes in many ways what one might expect for alternative stable states, which emerge when you have particular types of feedback loops. So all of this suggests that to understand what's going on in terms of within host dynamics, taking a dynamical perspective is going to be potentially very useful. It's really going to help us tease apart what's going on. But of course, the way I framed it here is incredibly abstract and incredibly high level. And so a natural question is, can we go a little deeper? And I'm going to talk through one way one might do that using the example of malaria, using some experimental data from mice. And what happens when you inject malaria into a mouse is that the parasite invades red blood cells. The parasite replicates inside the red blood cells and then bursts the red blood cells. And then the new merozoites, they're called, invade new red blood cells, replicate inside them, and the cycle repeats. At the same time, of course, new red blood cells are being, in some sense, born into the population, or at least are appearing, and the immune system is doing something, some mysterious thing where it's destroying things at different pieces and at different times. We don't see all of this incredibly nuanced biology. What we often see is simply numbers of uninfected red blood cells through time and numbers of infected red blood cells through time. So the uninfected red blood cells sort of collapse as the parasitemia goes up because the parasite is exploding red blood cells, and the mice become very anemic, and then they sort of recover as the parasites fall to low levels again. So you can think of two extreme ways you could model this and model the role of the immune system. You could do the sort of set of tools developed by Alan Perelson and Rustam Antier and others, where you write down 3 partial differential equations that are essentially designed to describe the whole world. And it's beautiful and it's very analytically constrained, and it has given us some incredibly remarkable insights, for example, into treatment for HIV. At the other extreme, what you might do is take a much more systems immunology approach, where you try and write down every possible set of interactions between every set of effector cells, laying down all the cytokines, and you end up with this incredible hairball network structure, which again can be very powerful for trying to understand what's going on, but is incredibly complicated and quite hard to work with. So on the one hand, we have this kind of extremely formal simplicity, and on the other hand, we have this complicated hairball. And we were interested in asking, is there something we can do sort of between the two? In particular, in this question of the evolution of thinking about how malaria spreads inside mice. And we were assisted by this in the fact that there's this very peculiar feature of malaria inside mice, which is that the bursting of red blood cells that I described following replication inside red blood cells happens in a 20-minute window around midnight, at least according to my empirical colleague's description, which is super weird. And we wrote a paper speculating as to how this might have evolved, which is one of the most fun papers to write, called the Cinderella syndrome. But what it does for us here is that it essentially allows us to say that we're working in discrete time. You know, they're moving from day to day, from midnight to midnight, in terms of how these dynamics unfold. So we can write down a relatively simple equation to describe how these dynamics are unfolding inside mice, where the number of infected red blood cells at T plus delta, where delta here is a day, is going to be an encounter defined by an encounter between infected red blood cells, uninfected red blood cells, the infected red blood cells turning into merozoites, so the burst size, the invasion rate of the uninfected red blood cells, and then loss to immunity. So this composite figure, it's phenomenological. It's not sort of this detailed analytical breakdown of what's going on, but it might be able to capture an awful lot of what the key processes are. And the lovely thing about this sort of expression is that you can simply log it and fit it using a regression. And we have numbers for the ITs, the IT plus deltas, the STs. And what we can fill in is the numbers around the transmission term there, the effective transmission term. And so if you do that, so this is from experiments that used 4 different inocular sizes, from low doses in black to very high doses in blue. And what I've shown across time is estimates of this number that is the composite of burst size, invasion rate, and loss to immunity. And what you see is that you start off sometimes increasing in the effective propagation number, but then it declines, presumably as the immune system kicks into gear and the ability of merozoites to invade new red blood cells is diminished, or the burst size is diminished. We don't know exactly what's going on, but presumably that's part of this trajectory. And the really nice thing here is, you know, we have this complicated process going on. We now have at least a window onto what's happening. And there are all sorts of really intriguing patterns, such as if you look at those the first couple of days following the invasion, following the introduction of malaria into this mouse, what you see is that log early abundance, so we've introduced different doses, right, translates into the saturating curve of the within host effective reproductive value, so the RE, which looks a lot like a type 2 functional response. So in some sense, at high density, it looks like the parasites are able to overwhelm the immune system. And this speaks to the evolution of this system, because we know that one of the way the malaria biology works is that the parasite moves to the liver and then replicates to reasonably high levels in the liver. This would be a sensible strategy. Of course, we don't know that this is necessarily why this is happening, but at least would be a sensible strategy if you're thinking that you're in this world where you kind of have an advantage from masting to overwhelm the immune system, like trees produce tons of seeds at some time. Similarly, if you look at the way in which you can back out from this math of the effective propagation number, you can back out what immune efficacy must be. So what the proportion of infected cells that must be removed by the immune system has to be to give you the dynamics that you see, having accounted for this effective propagation number, and assuming it's a constant number through time. And what you find there is that this very curious pattern. So at the top rows of this plot show small numbers of starting parasites, and the lower rows show large numbers of starting parasites. And as the number of parasites goes up, the initial inocular goes up, then the immune system responds faster. So the red comes earlier on the x-axis, but it also seems to forget more. So the sort of the green areas, there's this recrudescences that we observe for high doses. So it seems like in some ways, lower doses are more slowly responded to, but result in more effective memory. And there's a really interesting set of questions there about how memory will evolve, how we can think about magnitude of dose and the evolution of immune memory in the face of different doses, and how the handoff between innate and adaptive immunity might happen, which is sort of quite pertinent at the moment when people think about the different regimes that were inadvertently trialed, for example, for the Oxford vaccine. So in some sense, what I've tried to do is lay out how one might use semi-mechanistic models to show the kind of shadows on the wall cast by the immune systems. And then you can lay out these sorts of scaffolds for asking about immune system evolution. And I think that really the inherently dynamic nature of this system means that these sorts of time series analyses are going to be extremely powerful. Of course, we won't always be so lucky to have this beautiful discrete time biology to help us, but leveraging those sorts of time series, which are beginning to come out for SARS-CoV-2 and exist for a number of other pathogens, and being open to sort of moving between phenomenological and truly mechanistic framings. So a sort of semi-mechanistic approach is going to be very powerful. So that's building from the within host scale up in some sense. Sorry, oops. At the other end of the scale, so one could also ask questions about what's happening so this is sort of building from the within host scale, thinking about what the signature of immunity might be. We can also, of course, start at the population scale. And again, starting with SARS-CoV-2, one of the most notable features is really distinct patterns over sex and age. So this shows numbers of hospitalizations, ICU, and deaths for males in blue and females in green, indicating the very clear increase in the numbers of hospitalizations, et cetera, as a function of age, which we're all very familiar with, but also the excess in males that has been observed. This is actually data in collaboration with Santé Publique France and Henri and others. So we wanted to try and think about what this might tell us about immune system functioning and more broadly. And to do this, we laid it in the context of what we knew about other pathogens and other sources of morbidity or mortality. So what I've shown here is the relative risk of hospitalization against the risk in 60-year-olds for influenza and for all-cause mortality. And this is a very classic pattern. It's often referred to as a sort of bathtub shape in demography, where there's very high risks early in life that then decline and fall to a sort of plateau for a while and then come up at late ages. So this is not in any way unexpected. And what we can do then is lay on top of this what the patterns look like relative to 60-year-olds for SARS-CoV-2, but also for MERS. So MERS is Middle Eastern Respiratory Syndrome, and it's also a coronavirus. It's also a novel emergent coronavirus. And if we plot those out on the same graph, what we see is this. So it's very striking. The pattern is very broadly the same, but it seems to start increasing at a much lower age, which is not how we usually think of this, but it aligns broadly with what all good evolutionary biologists know, which is that senescence should kick in pretty much at puberty and then start going up. So based on this, one might infer that it could be that the flattening that we see in influenza and for all-cause hospitalization might be driven by protective adaptive immunity. So that might just be buffering out this lower age risk for those sorts of infections. Or it could be that these coronaviruses, they use this ACE2 receptor, which is a central part of the renin-angiotensin system, which is very linked to all sorts of things and all sorts of aspects of homeostasis. So it could be that there's something about the coronaviruses themselves that puts you in a very sensitive position relative to immunosenescence. So it's not so much that, that we are buffered relative to other pathogens. We know that we will all have had we're likely to have had several bouts of influenza by the time we reach puberty. So we're likely to have adaptive immunity to circulating seasonal flu. But it could also be the fact that there is this greater sensitivity just because of the biology of coronaviruses. So that's the pattern across age. Can we turn to the pattern across sex to see if there might be something going on there as well? And so what I'm going to show you here is the relative risk of hospitalization in males versus females, where values below 1 are going to indicate higher in females and values above 1 higher in males. And this is what you see. So SARS is shown in red, influenza in blue, and all cause in gray. And there's this very classic pattern well known to anyone who thinks about influenza, which is that females suffer greater morbidity and mortality between the ages of puberty and more or less menopause in influenza. And we also see something quite similar in SARS, except that the impacts go deeper for younger women and come up earlier and transition to being a male burden also at earlier ages. So what can we infer about mechanism here? Typically, when we think about sex differences in immune function, male bias is associated with a weaker immune response leading to pathogen damage, whereas female bias is associated with a stronger immune response leading to immunopathology. If we think that SARS and influenza are in some degree comparable, and we think that there's something about adaptive immunity going on, we might then infer that adaptive immunity is helping by sort of reducing these massive immunopathological responses in females, and therefore, resulting in this lower burden in influenza relative to SARS. CoV-2, I should say CoV-2, apologize. So taking a 1,000-mile step back again as an evolutionary biologist, the sort of classic trope, if you're a classic evolutionary biologist, is that males are the weaker sex. But this also indicates that it's a little more complicated than that. Males are the weaker sex, but there is a danger of immunity. It's not just that there's a cost of having a less functional immune system. You're seeing this pattern where at certain ages, females are also paying a cost of this higher immunity, apparently. And it really isn't, I think, so simple as to say that there is simply this males are the weaker sex type effect. There's a lot more going on. And in particular, it's not clear to me, given that there's clearly this balance of costs. You can be at risk of infection or you can be at risk of immunopathology, why you would end up with females tending towards protection against infection and males being more protected against immunopathology. And so some of the work I've done with Andrew Graham in particular to try and pick this apart is to think about how the immune system works in a very general sense and to break apart the functioning into two pieces. So if you build a really simplistic cartoon of how the immune system works, you can say that one thing it has to do is discriminate between self and non-self. And so it has to sort of draw a line that separates these two histograms, which is associated with a risk of false positives and also false negatives. So false positives is when you react to yourself, and false negatives is when you miss a pathogen. And everything, if you try and draw a kind of cartoon across the literature of how the cellular biology of the immune system works in females versus males, it looks like the female immune system tends towards higher, what epidemiologists would call sensitivity. So more likelihood of false positives and reduced likelihood of false negatives, because there are more and more efficient antibody presenting cells, higher titers in response to vaccination, a whole suite of different characteristics. So that's one trade-off one can imagine between these false positives and false negatives. There's of course another relatively simple trade-off again in building this super simple depiction of how the immune system functions. And that is in thinking about the magnitude of the response. And you can imagine that you might deploy a huge number of immune effectors, which would be very good at reducing your hazard or your risk of death associated with infection, but might increase your risk of death associated with immunopathology. So there will be a trade-off between those two ends of the extreme. And again, if you try and read through the literature on cellular immunity and sort of within host dynamics in males versus females, It looks like males, they have more natural killer cells. They have sort of a higher ratio of CD8 T cells to CD4 T cells. It looks like they're more on the end of the immunopathology. So a greater response, a less disease reactive response. Sorry, a greater response in controlling the disease. And if you take, one doesn't a priori know how this should fall out. But if you put these two trade-offs together and look at what the sort of fitness landscape might look like in very simple terms, using just a very simple expression for the hazard, what you find is that a landscape which unsurprisingly has two optima on it, one corresponding to high sensitivity, so many false positives, but a relatively small response in terms of, so you know, if you're triggering your immune response all the time, you probably don't want to have this massive response because it's sometimes wrong. And vice versa. So it's not that surprising that you end up with two optima if you lay it out this way. What is surprising is that there's still no particular reason that you would get females sitting on one peak, the peak that corresponds to this more sensitive, more triggered immune system, and males sitting on the other. So I spent some time trying to think about why you would end up with one of the two sexes on one of the peaks versus the other. And again, the trope as an evolutionary biologist is to think about how males might be channeling resources away from immunity towards, you know, peacock tails and fancy displays of one kind and another. And if you put in those sorts of costs, so say, for example, it could be sloppier discrimination on the discrimination axis, or it could be a smaller investment in immune responsiveness on the magnitude of response trade-off, you just don't get less sensitive males. And I really do hate the fact that the word is sensitive in the epidemiology literature. But it just doesn't give you the landscape that seems to match what we see in the literature. So that doesn't seem to be the answer. You can't pull out the pattern that we expect from immune trade-offs alone. You can start laying this into sort of very classic life history framework. So survivorship times fertility, the sum of the product of the two would give you some layer of fitness. And one might expect that pregnancy could result in higher mortality, higher infection-associated mortality. You can lay in all these sorts of trade-offs. And you can also layer in the fact that, and this is work that Andrea Graham did, that we expect a higher sensitivity, which is effectively higher responsiveness to self as well as non-self, could result in miscarriage. And in fact, it does seem to result in miscarriage in some sheep females, but those females that are also better protected against infection. So you can put all these trade-offs together, and there isn't a really convincing picture as to why you'd still end up with females having greater sensitivity. And I forgot to mention that one of the reasons that this greater sensitivity in females, one of the lines of evidence for it is that 80% of autoimmunity is concentrated in females, which is a really stunning percentage and corresponds to this self-reactivity. And so sort of from there, one thing I did was it's clear that this model is ridiculously simple in terms of how things actually work. And one of the obvious simplifications and differences between males and females is in terms of the transfer of maternal immunity, which is linked to adaptive immunity. And you can build a relatively simple sort of individual-based model to capture this and show that indeed, if there is important survival benefits associated with your mother having had a diverse repertoire, then you can end up with a conclusion that females should evolve towards higher sensitivity and males would end up on the other peak, the greater specificity peak. So,
sorry, I am going to interrupt. Your time is kind of up. So if
okay, yeah,
it's
sure.
It's super interesting, but if you could.
No, of course.
Thanks a lot.
Thanks very much. Sorry,
Oh, okay. So I guess I was just okay. So sorry if I cut your conclusion. So now, feel free to again write your